Welcome back to the third lecture in our chapter 18. So now we're going to talk about leukocytes. So these are your white blood cells. Leukocytes are different from erythrocytes in that they contain all of their organelles and the nucleus, so they can survive longer, they can fix damage. They are motile, so they can move around. They're not just floating through the blood. They can actually move to areas where there's damaged tissue or where there is some infection or war occurring. And their major job is to function in protection of the body. Okay. Oh, there we go. All right, so here is, I told you that they can move to the site of an infection. So here we have damaged tissue of the body. Damaged tissue as well as pathogens can release chemicals. These chemicals enter our blood, and our blood cells, our white blood cells, can respond by moving towards the damaged tissue in a positive chemotactic mechanism. Once they get there, they're going to squeeze through the walls of the blood vessels. The term for squeezing through the walls is called diapedesis. They move in, and then they're going to fight off the infection. They're either going to phagocytize the material, phagocytize the damaged tissue, or they might stop the bleeding if they're platelets. So what are the white blood cells? Really, we have granulocytes and agranulocytes. I do have platelets on here, but platelets aren't really part of the white blood cells. They're a cell all of their own. So we're going to talk about them separately. Granulocytes are your phils. And so we have neutrophils, eosinophils, and basophils. They all contain large granules that you can see. Whereas agranulocytes, lymphocytes, and monocytes do not have those granules. So here we can see the different white blood cells. Here we have basophils. You can see all those granules. Here's eosinophils. You can see those granules. Here's the neutrophil. You can see the granules. Here's a monocyte. And then here's a lymphocyte. No granules in these structures. The purple structure here, 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 and here, and you can kind of see it in there. That's your nucleus, okay? So each of these white blood cells has their own major job. Neutrophils, which are the most common of your white blood cells, typically become even more common during bacterial infections. Neutrophils are phagocytic cells. They're your common phagocytizers. So they're the first responders to an infection or to damaged tissue. Neutrophils will phagocytize material, but then they die. Eosinophils are also able to phagocytize materials. They don't do it like neutrophils, so they don't move to sites where there's bacteria and just start phagocytizing. They become common during either an allergic reaction or a parasitic infection. So parasitic worm infection, I should say. So worms in the digestive system. Eosinophils release an antihistamine, which stops inflammatory responses. They also can phagocytize certain worm pathogens. And then basophils, these are the least common of all of our white blood cells. Basophils contain two different chemicals in their granules. They have histamine, so eosinophils have antihistamine. Basophils have histamine. Histamine is used to cause the swelling and the redness, soreness during an allergic reaction. So when you have that, the red itchy eyes or the red bumps on your skin when you touch like poison ivy or whatever, that is due to the basophils releasing these chemicals. They also release histamine and heparin. Heparin is an anti coagulant. So it causes blood to be a little bit leakier. Your blood vessels become more leaky, and so blood can move to sites, which is what causes that inflammatory response, the raised red bumps. Then we have lymphocytes. We have different types of lymphocytes. So we have, and so these are your lymphocytes. Lymphocytes are very easy to identify because the nucleus takes up almost the entire cell. Lymphocytes are going to either function in specific immunity, B and T lymphocytes, or in non-specific functions, your natural killer cells. And so lymphocytes are going to be talked about more during the immune system. But for now, when you think of lymphocytes, think of long-term infections like viral infections or chronic bacterial infections are when they become really common. And then we have monocytes. So monocytes here are your largest of your white blood cells. And when they get activated, they become macrophages. And they have these little projections off of their plasma membrane that allow them to phagocytize a lot of material. Macrophages are like your professional phagocytes. So whereas neutrophils will digest or will take in bacterial cell, and once they have taken in a cell or two cells, then they basically can no longer function. Monocytes can phagocytize multiple cells, lots of cells. They don't get damaged. And so when monocytes do this, they can actually then present these pathogens on the cell surface, which can activate lymphocytes. So monocytes are very important in both nonspecific and specific immunity. They kind of work together. So let's look at some disorders associated with our leukocytes. Lymphocytosis is an increased number of lymphocytes. Lymphocytopenia is decreased number of lymphocytes. Leukocytosis is just an increased number of leukocytes, while leukopenia is decreased leukocyte count. Lymphocytosis is often associated with viral infections or chronic bacterial infections. Leukocytosis is typically caused by infections that are either viral or bacterial, so you have just increased number of white blood cells in general, versus lymphocytosis is increased lymphocytes, so those specific defenders. Leukopenia, that decreased white blood cell count, is often associated with extreme stress or certain types of cancers, whereas lymphocytopenia, decreased number of lymphocytes, is associated with HIV, so a viral infection, or certain types of leukemia. There we go. So what is leukemia? It's cancer of the hemopoietic tissue. And it causes a high number of leukocytes to be produced, but these leukocytes are not functional. So they're like immature leukocytes that don't have a job. And because of that, we have lower than normal numbers of good white blood cells. Myeloid leukemia is uncontrolled granulocyte production, while lymphoid leukemia is uncontrolled lymphocyte production. The last type of cell then is our platelet. So platelets are derived from a larger cell, or the last, cell type, I'll just say cell type, is derived from a larger cell called a megakaryocyte. Platelets aren't actual cells, they're pieces of a cell. So we have this myeloid stem cell that differentiates into a megakaryoblast, an immature cell that eventually becomes a megakaryocyte. The megakaryocyte then has these long plasma extensions, and these plasma extensions go into, so this is in the bone marrow, these extensions move into the blood, and pieces of the megakaryocyte are broken off, forming those platelets. So platelets function in blood clotting or hemostasis. And so if you have an injury to a blood vessel, this is going to initiate hemostasis. There are 3 basic steps to hemostasis. We have the first is your vascular spasm. Then you have a platelet plug formation, and then finally you have coagulation. So here it shows 4 steps because you have to have an injury, but that's not really a step of the hemostatic mechanism. That's something that happened that brought our body out of homeostasis. So from here down is actually the hemostasis process, okay? Oh, no, I want to talk a little bit longer. I'm sorry. So when you have damage to a blood vessel, this can be external damage. So like you cut yourself when you were dicing potatoes, you nicked your finger and you started bleeding. Or it can be internal where something inside the blood vessel damages the blood vessel and so you start bleeding internally. In both cases, the initial function or the initial occurrence is going to be these platelets are going to be moving along, and we have our blood vessels are coated with an it's basically a chemical that keeps our cells from sticking, okay? So that we don't get these buildups of, platelets or anything else. You don't want a blood clot in an unopened, undamaged vessel. But when you have damage to the vessel, then you have extensions of the plasma membrane that are available that don't have that special coating on them. And platelets are going to recognize that. So the first thing that happens is you have a vascular spasm. So when you have damage, the blood vessel itself is going to spasm, constrict, so that you don't lose as much blood from the damaged tissue. Then platelets are going to move to the site where they can identify the collagen fibers in the blood vessel. When they do that, they release chemicals. So when the first platelets attach, they release chemicals, and these chemicals activate other platelets or stimulate other platelets to move to that site. So this is a positive feedback. The more platelets that attach, the more platelets that are going to come. The more chemicals are released, the more platelets are stimulated to move to that site until we have a platelet plug. So from there, we have coagulation occur. So we have these protein fibers called fibrinogen. Fibrinogen is inactive in our blood plasma. It's produced by the liver and it just runs through our system. It's inactive. So when the protein is acted on by thrombin, the fibrinogen is converted to fibrin. And fibrin are the active proteins in the blood clot. So this can occur through either intrinsic or an extrinsic path. So extrinsic would be like the nicking of the finger. Intrinsic is the damage to the blood vessel. But in both cases, we have a cascade of protein factors. So all of these different factors here, this one is factor 12, and yeah, 12. This is factor 10. This is factor 9. Factor 12 is converted to factor 12A, which converts factor 11 into factor 11A, which converts factor 9 into factor 9A. So do you see how that works? These, and I don't care if you know all of these. These are just clotting factors that are produced also by the liver. Over here we have trauma that's external. So we have factor 3 that converts factor 8 into factor 8, I think A again. In both cases, though, we eventually lead to the common pathway where eventually prothrombin is converted into thrombin, and thrombin is what is needed to convert fibrinogen into fibrin, and fibrin is what forms the meshwork of that blood clot. And so then you have that nice tight blood clot, which has some red blood cells in it, a little bit of white blood cells in it, those platelets, and it sits there over the damaged area until the damage has been healed from the inside out. And then that clot will fall off as a scab falls off. So some disorders associated with platelets, we have hemophilia. This is a genetic disorder where you don't have certain factors. So again, this is why it's important to kind of see that there are a lot of different factors here. We have a lot of factors that are needed to convert eventually the prothrombin into thrombin. If any of this cascade is stopped, then you're not going to produce that thrombin. And so in hemophilia, one of the most common forms is hemophilia A or B. These are X-linked recessive, so they typically are seen in males because males only have one X chromosome. Females can be carriers. They can also have hemophilia, but they typically are carriers. And in this case, you have either a deficiency in clotting factor 8 or clotting factor 9. And so people with hemophilia don't clot properly, so they bleed out more. And when that happens, they're going to, if they bleed out more, they're not going to have the oxygen carrying capacity. If they bleed out too much, they could actually die. So things like a bloody nose can make you run to the ER. Today, we have a lot of different medicines that can be used to help clot a typical, if you cut yourself and you have hemophilia, they have clotting chemicals that you can just put on that damaged skin or that damage, and it'll clot that area for you. But in the 1800s, in the 1700s, they didn't. And so a lot of times people would die because they have this disorder. Another disorder is thrombocytopenia. So this is where you don't have enough platelets. So in this disorder, you might end up having a lot of bruising because any damage internal that normally your platelets are going to fix leads to leakage of a lot of blood, which the leakage of blood is what causes the bruising that you see here. HyperCoagulation is another problem, and it can lead to a lot of serious issues. So hypercoagulation is when you end up producing clots inside of an undamaged blood vessel. And so here you see a thrombus. A thrombus is a blood clot inside an unbroken blood vessel. If that thrombus dislodges and starts moving, so here you can see part of that thrombus dislodges, and so now you have this little piece here. This is an embolus. Emboluses are very, very dangerous because they can clog up some of our major arteries. And when that happens, it can lead to the inability to breathe properly, the inability to get oxygen to our tissues properly, and this can lead to death. I'm going to stop here on this video. The last thing we'll talk about are blood types, which is always fun to do. Bye.